Hello, am I audible?
Yes, ma'am.
Yeah, okay. Is my presentation visible?
Yes, ma'am, it is visible.
Okay, let me know anytime if it's not visible or if I'm not audible. Uh,
Sure, ma'am.
okay. Hi, welcome to Cell Culture Technologies Week 6 live session. This is an NPTEL course and the course instructor is Professor Manik Das from IIT Kanpur. I'm Sumati Raviraj from University of Hyderabad. So this week, the module was cell separation and in vitro myelination cell culture mode. So you have 1 to 5 modules for the same topic. So right now we will be solving the weekly assignment for this. So if you know the answer, just let me know and answer the question. If not, we will go further and start discussing the answer. Okay. So the first question is astroglia. What is astroglia? That is, and what does it do? Does it help in maintaining cellular homeostasis, or are they immune cells of the brain, or they form myelin sheath, or none of the above? If you know the answer, you can answer it.
It is to maintain the cellular homeostasis.
Yeah, very good. So, astroglial cells are helpful in maintaining the cellular homeostasis. One second. Yeah, so the answer is A, maintain cellular homeostasis. So what exactly is this? What is that homeostasis that these astroglial cells make? Are There are different ways in which it will maintain the homeostasis. One is molecular homeostasis, that is ion homeostasis, like the potassium ions, chloride ions, calcium ions. These are very important to maintain the cellular ionic balance. So these cells help in maintaining this, and in course of maintaining ion balance, it will also help in regulation of the pH. And then in turn also the water transport also will be maintained based on the concentration of these ions within the cell and outside the cell into the matrix, etc. And it is also a neurotransmitter homeostasis maintainer. So neurotransmitters like glutamate, GABA, adenosine, monoamines, all these also it will help in maintaining the uh, homeostasis or the concentration of these neurotransmitters. And then it will also help in systemic homeostasis, that is in chemosensing, like to sense the levels of oxygen or to sense the levels of carbon dioxide, higher levels or lower levels of glucose, sodium, etc. So this oxygen, carbon dioxide is essential for your cell to maintain in the cellular physiological condition. So CO2 will also help in your regulation of pH, and your glucose is required for your energy balance. So this glucose balance is generally based on your food intake. So depending on what type of food you eat or what how much amount is there, your glucose will be maintained depending on the food intake as well. And then it is also helpful in maintaining your sleep cycle. So your sleep homeostasis also will be maintained by your astroglia. Then organ homeostasis. So at a larger level, it will have a control over your blood brain barrier, and it is also helpful in operation of the lymphatic system. Then comes metabolic homeostasis, formation of neurogliovascular unit and glial vascular interface. So these are these two interfaces And here you can have the regulation of your blood flow, your metabolic support from other signaling, and your glycogen synthesis, storage, etc. So your neurogliovascular unit and your glial vascular interface. Then it also helps in cellular and network homeostasis. That is your neurogenesis, your uh, during development or during course of information processing or something. When you have there is a need for neuronal generation. So your neurogenesis also is maintained by your astroglial cells and its network of homeostasis, your neuronal development and your neuronal guidance. So, as I said, in your development, during the developmental stages of your brain, your neuronal cells, when they have to, start developing, and where is the fate of that cell going, in which area the neurons have to develop more, which area less. So, these kind of guidance signals also is given by this. At the same time, it also defines the cytoarchitecture of the central nervous system. So, your central nervous system is a huge network. So, the architecture of this is very important because it will help in sending signals to different parts of your body. So, the central nervous system architecture is also maintained. Then, synaptogenesis, synaptic maintenance, and synaptic elimination. So, your synaptic plasticity, that is the dynamic nature of your synapses, is also maintained with your astroglia. So, so major of your complete cellular homeostasis is under the control of astroglia. So your synapses. So in the synapses there is uh, tiny spaces, and then the communication with each other, your cognitive, your learning cells, all these are coming under your synapses. And presynaptic, postsynaptic signals from your axon end, all these electrical impulses, etc., will also come under your synaptic maintenance and its plasticity. Okay, so the next is your microglia. So, what is microglia? What is the function of that? Is that also involved in maintaining cellular homeostasis, or are they the immune cells of the brain, or do they form the myelin sheath, or none of them are? What is the answer for this?
So they are immune cells of the brain.
Yes. So, microglia are the immune cells of the brain. So, they are like macrophage like cells which help in the defense, immune defense of your brain. So, this microglia is also involved in major of your neuronal diseases and disorders like your Alzheimer's, your dementia, your Parkinson's disease, etc. So here you can see in these other sorts of neuronal impairment or diseases like your tau related diseases like we discussed in the previous sessions, your hippocampal and cortical neurodegeneration occurs in this, and then your plaque formation is also triggered by your microglial activation. So there are other genes that is also involved in this, like your TREM2, your NLRP3. These are all major genes that are involved in your inflammation and pro-inflammation. So all these genes are also activated by the microglial signalings. So your ApoE expression. These are major immunological proteins which are under the regulation of your microglial cells. Then in your frontotemporal dementia. So here also your tau comes into play, your TDP43 comes into play. And here you can see this is your neuron, this is your microglial cell. You can see the shape of the microglial cell. It has a lot of processes emerging from the cell body. It has this projections kind of thing. It helps in the movement of these cells. So most of your immune cells have this ability of this movement and these kind of projections which help them. So you can see it has a macrophage like structure and even more defined processes from the cell body. So so your tau containing tangles are also triggered by your mitochondrial response. So microglial defect will also lead to release of your secretory proteins like your progranulin, etc. And then your mutations in those genes which are related, like your TREM2, will also lead to these kind of dementia like diseases. And then in Parkinson's disease, your intracellular alpha nuclein protein will aggregate and Lewy bodies will be formed. So then there will be neurodegeneration. So your microglia will get activated here, and the neuronal dysfunction will send signal for your microglial cells to get activated and start the immune response over there. Then your TLR2, TLR4, your toll-like receptors, these are also very important for the immunological signals. So all these are also present in your microglial cells with MHC2 class upregulation, and this will lead to your inflammation or your injury in the neurons during Parkinson's disease. So your microglial cells are very important in maintenance of your immune system of the brain, and if there is any injury or any sort of distortion in the proteins or any sort of accumulation of proteins which is not supposed to happen, then your microglial cells get activated. So here you can see this is a Venn diagram. So this is your microglial cell and this is your normal blood monocyte. So your monocyte is also similar. Your microglial cell is similar to your macrophage like I mentioned before. So your monocyte stage is similar to your embryonic microglial progenitor. So what happens is generally your monocyte is a dormant state where it does not have major immunological response, but once there is injury, your monocyte will get differentiated into your peripheral macrophage, and this macrophage is your major immune cell which will come into your pathogen invasion or any kind of injury, and it will come and act over in that place. So in the similar way, your microglial cell will also be acting like a macrophage in your brain. So you can see these are the genes that are expressed by your microglial cells, which is in this blue circle, and this yellow circle which you can see are the genes which are common to both your microglial and your macrophage cells. So these genes like CD68, F480, CX3CR1, IBA1, all these are immunologically related genes. So these are expressed in both macrophage and microglia. So that means that this shows the similarity between your microglia and your macrophage cells. And then you can also see in the red that there are leukocytes. So in this leukocytes, you have your CD45 and CD11B markers. So these markers are also present in your microglia. So this is basically to say the similarity or resemblance between your immune cells, major blood immune cells, and your microglial cells. So coming to the next question. Density gradient centrifugation utilizes a column with dash density from top to bottom. That is your order of density. A increasing, A decreasing, B increasing, C same, and D none of the above. So what is the answer for this?
It is in the increasing density.
It is your increasing density. From top to bottom, when you see it, the density will be increasing. From bottom to top, it is decreasing. So your highest density will come and settle at the bottom, right? Because there is more mass related to relative to the volume in that. So your density will be increasing from top to bottom. Your lowest density will be at the top and your highest density will be at the bottom. So what is density gradient centrifugation? It is a procedure for separating particles such as viruses or ribosomes or molecules such as DNA or even cells, different types of cells based on if there is a difference in the density of the cells, you can separate. So anything in which you have a mixture of either proteins or biological molecules or cells which differ in density from each other can be separated by your density gradient centrifugation. So you can separate particles in which the sample is placed on the preformed gradient. So like in the previously they asked what is the order of the gradient. So this gradient is a preformed gradient which is a sucrose or cesium chloride gradient. So in this sucrose gradient or cesium chloride gradient only, according to the different levels of the percentage of it, your molecules or your viruses or your any cellular component that you're trying to separate will settle at different uh, gradient area or different gradient line in your column. Upon centrifugation, either by rate zonal or equilibrium procedures, the macromolecules are banded, that is your different layers. It becomes, like I said, it will come and settle at different layers. So that is called banded in the gradient according to its density. And then they can be collected as pure fraction by taking up only that band from the entire gradient. So here you can see this is your column. Here you have your ficol. So your ficol is also your density gradient medium where it will have the higher density. So here they have shown an example of your blood samples where they are trying to separate the cells from your different layers of your blood. So in such a gradient which is already given in the sample, you add your blood in your column, you add your blood sample and then you centrifuge it at a particular RPM. So then what happens? The different components of your blood will get separated according to its density. So here you can see RBC and neutrophils have come at the bottommost layer. Then you have your ficol. Then on top of that you have your WBC layer, your white blood cells layer. And at the topmost layer you have your plasma and your CTCs. So here you can see this separation is based on the difference in their density. So your plasma and your CTC both have similar density. So they both come in a same layer. And then you have your WBC, then your RBC and neutrophils are also of the same gradient. So both of them are in the same layer or same band. And then you have your types of centrifugation. So there are different types for different applications. So your major density gradient centrifugations are of two basic types, that is your rate zonal centrifugation and your isopycnic centrifugation. So here you can see the different percentages. This is a real isolated form. So here you can see the difference in their density based on which they have been separated. So here you have your percentages, at what percentage which one is there. So you can see from top to bottom your density is increasing. So that the bottommost you have around 60% here, at the uh, top of that you have 40%, then you have 25%, then you have 17%, and since viruses are like very smaller molecules, they will come in your topmost layer. So now we spoke about the rate zonal and isopycnic, that are the two types of your density gradient centrifugation. So what is the difference between these two? So your rate zonal centrifugation is an analytical technique we can use to separate particles of a solution depending on the size but not the density. So in your isopycnic centrifugation, uh, we separate the particles depending upon the density but not the size. So here also, if you see, even though there is a density gradient in your rate zonal, they get separated based on their size, and in your isopycnic it is based on the density. So here the size and density is of your sample. So based on the size of the component in the sample, if it gets separated, it will be in the rate zonal centrifugation. And if your sample can get separated based on the density of the components in your sample, then it is isopycnic centrifugation. So you can remember this easily. If it is size, then it is rate zonal. If it is density of the components of the sample, it is isopycnic. Then what is the nature of the particle? So since it is getting separated by the size, so obviously there is a difference in the sizes of each component in your sample in the case of rate zonal. In the same way, in the case of isopycnic, because it is based on the density of the sample, the particles in your sample will have different densities from each other. So what is the effect of time is, this is time dependent and this is independent of your time. So based on what, so your time, whatever you take at particular step, it is very important in case of your rate zonal centrifugation. And because it is your size, so if you do it lesser than the stipulated time, then your particle did not have enough time according to its size to come at a particular zone. If you do it more than the stipulated time, that means your particle is now moving farther and farther from the zone, because it is your size, because your size is not as specific as your density. So depending on your time, the separation of this size related components will change. But in case of your density, once it has gone and settled at the appropriate or its complementary or its similar density layer or zone or band, then once it has settled in that, unless it is disturbed by an external source, it will not move even if the time taken is more or less than the stipulated time. So density gradient is more specific in case of isopycnic. And then what is the nature of gradient? Gradient nature. So the gradient nature is flat, the gradient nature is steep here. And what is the type of pellet formation? Here the pellets are formed, here no pellets are formed. And the application is for separation of cellular organelles, proteins, etc. And isopycnic is for the separation of your nucleic acids. So based on what type of experiment it is, what you want to separate, and what organelle you want or what component of your cell you want, you have to think about that and choose the type of centrifugation that you want to go. Then based on that, you can use your gradient differing in that and your protocols. So the major applications of your density gradient centrifugation is cell separation. Like you saw here, the plasma and WBC, RBC, all these are separated. So different types of cells with different densities you can separate. Then separating components of whole blood. So this is your whole blood sample in this experiment, in this example. You saw how the plasma was separated, WBC was separated. So here this is not a complete separation of your cell types and your components. This is your whole blood separation. So after this, you can even separate your CTCs again separately, your RBCs separately from your neutrophils, etc., using a more refined density gradient. Then you can also do purification of viral vectors. So your viral vectors are your carriers for your genes or your transfection. So they are your plasmids that you have, your retroviral vectors. So these vectors also, if they are in a mixture or if you want to isolate it from your different types of cells, you can use this kind of centrifugation method. Then separating bacteria from food. So if you have food contaminated with bacteria, you can also use the density gradient to separate your bacteria from your food. So because your food components will go at a particular density band based on their size and density, and your bacteria will be at your particular state, similar to how here you see the virus at the topmost layer. In the similar way, your bacteria can also be separated from a mixture, even from the food. So your other thing is your molecular weight distribution. Like you saw here in the red zonal, your particles are getting separated based on their size. So when such particles are getting separated on the size, uh, you can distinguish your molecular weight of different components in your mixture. So your distribution of different molecular weight components can also be analyzed and studied using the density gradient centrifugation, that is your rate zonal centrifugation. So if you have any doubt, you can stop me at any time and you can ask me. Okay. Um, so let's go to the next question. So in the next question, which of these is not a cell of the nervous system? Neuron, astroglia, Schwann cell, nephron. Which is not a cell of the nervous system? What is the answer?
Ma'am, it is nephron. I'm just okay with this fourth and fifth question. Can we skip to the sixth one, ma'am?
It's fine. We'll go through it. So yeah, this is a relatively simple answer. So it is your nephron. One second. Huh. So the answer is nephron. So what here I am trying to say is, what are your different types of your neuronal cells? So here particularly we are seeing different neuroglial cell types and their functions. So because in the previous questions we got your astroglia and your microglia, we can see that your central nervous system and your peripheral nervous system are there. And in your central nervous system you have astrocytes, you have ependymal cells and your oligodendrocytes along with your microglia. Whereas in your peripheral nervous system, you have your satellite cells and your Schwann cells. So your astrocytes, they maintain blood-brain barrier, controlling the levels of neurotransmitter around synapses, and they regulate ion and providing metabolic support. So this we saw in the first question, that the astrocytes, they maintain the synapses, they maintain the metabolic homeostasis of the cell. They also maintain your neurotransmitters, etc. So then comes your ependymal cells. Your ependymal cells line the spinal cord and ventricles of your brain, and they are
Excuse
also
me.
involved in producing cerebrospinal fluid. So your ependymal cells is important in maintaining your levels of cerebrospinal fluid, because this is very important to protect your protect your brain from shocks and pressure from external source also, and also to maintain the shape and structure of your brain. Next comes the oligodendrocytes. Oligodendrocytes are myelinated and they belong to the central nervous system, axons, and they provide a structural network and framework. And then comes your microglial cells. So your microglial cells are your brain's immune cells, and they help in removal of dead cells because they are macrophage like cells. They help in removing all your dead particles or your apoptotic type of cells, etc. And then your pathogens. So like how your macrophage is also involved in attacking your pathogens by identification your with your help of APC cells, with your MHC molecules. In the same way, like we saw in that Venn diagram, where your microglial cells also has your MHC class 2 markers. So this will also help the microglial cells in identification of pathogens, and all your pathogens and your dead debris around your brain will be phagocytosed and removed from the region. That is the function of your microglial cells. And in your peripheral nervous system, we have satellite cells and Schwann cells. So your satellite cells They look like this. They look like a satellite. That's why they're named satellite cells. And then they surround the neuron like satellites, like how satellites surround the planets. They surround your neuronal cell bodies in ganglia, and then they regulate the neurotransmitter levels. So, or your neurotransmitters, how much concentration it has to be supplied, that is regulated by your satellite cells. One second. Huh. Next comes your Schwann cells. Your Schwann cells are also your myelinated neurons in your peripheral nervous system, and they help in maintenance and regeneration of neurons after injury. So you have your pro-inflammatory and anti-inflammatory cells. So in your and then in macrophages also you have your M1 and M2 type. One is involved in pro-inflammation, where it causes destruction to the region of injury in clearance and attacking the pathogens. And then the other one is whatever destruction has been made, that region has to be now repaired. So your other type of macrophages will help in repairing of the tissue. So in the similar way, your Schwann cells also will help in regeneration of your neurons after the injury is caused. And they are all known to support your uh, peripheral nervous neurons, and they are located in your axon localization is seen. And then in case of inflammation, what happens, your diseases or neuropathic pain or your polyneuropathies, etc., Schwann cells are involved in the development of this kind of nervous system related to inflammation. They also help in maintaining the structure of your this axon with their myelin sheath. Okay, so this is your different types of cells. Your next question is, which of these is recommended density gradient medium? What is the answer for this?
It is OptiPrep.
Yeah, so this is OptiPrep, that is your C. So your OptiPrep is a density gradient medium that is used to isolate biological particles in vitro, that is by a density gradient centrifugation method. And then it is sterile, and it is endotoxin tested solution of 60% iodoxanol. So your iodoxanol is the major component. You can see it is 60% in water and with a density of 1.320 grams per ml. So what are the major applications that is the OptiPrep is used is your cell isolation, your viral particle purification, the dendritic cell research, cell sourcing, that is nothing but cell isolation, and then the cell differentiation and maturation. In all these kind of experiments, you can use your OptiPrep medium. And then OptiPrep medium is biocompatible, that is, it is non-ionic and non-toxic. This is very important because your ion if your ion balance is impaired by the medium, then it will affect the impulses and the signalings if you are especially working in case of cell differentiation and maturation. Then it also has low osmotic pressure so that it doesn't uh, give any sensitive impulses to your cells. And then it is biologically inert. It does not have biologically inert in the sense it does not have any property, any antigenic property which might cause any stimulus in your cells to release certain secretory immune response. So it does not have any such sort of biological antigens in it, and it does not interfere with the assays for marker enzymes. So when you're doing particular marker analysis like your ELISA or your flow cytometry, etc., where you use particular protein tag to identify certain cells, or particular tags to identify certain proteins and certain enzymes. If there is any biological component in this medium that might affect your reaction. So all these marker enzyme assays will have a reaction that occurs to identify the either qualitative or quantitative, either the type of enzyme or the amount of the enzyme. So if there is any component that might affect this reaction or might add to this reaction, should not be present in the medium. So opti prep medium is biologically inert in this way. Then it also forms spontaneous gradients that are easier to prepare than sucrose. So in the previously we saw that usually sucrose and cesium chloride was used for a long time in this density gradient centrifugation. So this OptiEM is much easier and better version of your sucrose because it helps in easier gradient formation and it is pretty quick than other gradients to settle down. And your opti prep is stable for 3 years if it is kept sterile, and it should be protected from sunlight because it's sensitive to light, and it should be stored between 4 degrees and 24 degrees. So why the storage information I have kept is whatever reagents that we use, you have to always check the catalog and you have to see first where you have to store it and in which form, at what temperature is required in the storage. Certain reagents might be desiccated or certain reagents might be in powdered form, and then you will reconstitute it in PBS or autoclaved water. So in that case, your desiccated and your powdered form will have a different storage temperature, and then your reconstituted one will have a different storage temperature. So if you don't look into these particular temperatures, your product will degrade or lose its activity. So it is very important to always check the storage temperature of any reagent that you purchase. Okay. So the next question is based on your answer in question. Yeah.
Ah, in the first question, what are the rest three answers, ma'am? Those are also the density gradient, or what are those? Yes.
No, no, those are just some other options they have given. OptiPrep is actually the name of that reagent which we use.
Okay. So the rest three are not a density gradient
No,
anymore.
no, rest three are not used in density gradient.
Okay. Thank you.
Then, based on your answer in question 5, that is, your answer in question 5 was your OptiPrep. So, based on this, what is the density of recommended density gradient medium? What is the density? Is it 0.32 mg per ml or 0.32 grams per ml, 1.32 mg per ml, or 1.32 grams per ml? What is the answer?
It is 1.32 gram per ml.
Yeah. So this is 1.32 grams per ml. So like I was saying in the previous, you have to check all the information of any reagent that you buy. Even if you have it in your protocol, after purchasing, you have to see. Sometimes it might differ from company to company also. So these kind of features, if you look online itself, even if you don't have the bottle or if you have transferred the bottle and lost the actual bottle, so you have to always remember the catalog number. So in the catalog number, if you put it on Google, you will get the web page for the product, and there they will have this first basic features that is required to be known where when having this product with you or when using this product with you. So in case of this OptiPrep, these were the features given in the product information. So the density is also measured here. It is 1.32 plus or minus 0.001 gram per ml. So this plus or minus is your error range. So any analysis that you do will always have an error bar or error range. So you should also be sure that your error range is not very high. So if your error range is very high, then this density might be different and there might be some issues while using, and then you cannot be sure whether it was a issue with the product or issue in your protocol or in your sample. So here you can see the error range is very small. It is plus or minus 0.001. So this is a good analytically tested product. So then the osmolality is also given, that is 170 plus or minus 50 milliosmoles, and then the endotoxin levels are also given less than 1 EU per ml. So your endotoxin levels and all this are also very important because as they have said that it is biologically inert. So there should not be any foreign object in this. There should not be any toxic substances, or there should not be any enzymatically active products, components in this, so that it should not interfere in your assay methods when you're performing your experiment. Then the stability, like it was mentioned before also, it is stable for 3 years if kept sterile and protected from sunlight, and storage is 4 to 24 degrees. Then, so this is the page you can see and visit if you want to know more about the preparation methods or the components in this OptiPrep medium, and how it is prepared, what is the structure of it, and what components are used, and how you can, put it for different applications in your experiments. So here you can see that it was mentioned the iodoxanol levels are 60% in this in water. So how they have developed this will also be given in this. And iodoxanol is a non-ionic, non-toxic to the cells. So here they have also given, particularly for particular products, they will also mention what applications this is more useful for. And if this, whatever application you're looking for, if there is a product that is more ideal than this product, that will also be mentioned in the web page. So here in the applications they have given, it is used for isolation of PM from the cardiac muscle by flotation. So they will tell you in which method you can use it for what particular isolation. So then isolation of PM and domains using uh, cationic colloidal silica. Then isolation of PM by flotation through continuous gradient at 16-hour spin. So why they are mentioning this particular things, 16-hour spin, this medium, and this particular uh, column or something, is only in that it will function properly for that particular experiment. So if you want to isolate PM from your particular flotation method, you have to use a continuous gradient for 16-hour spin. Otherwise, it will not work. So these kind of tiny details are very important when you're doing such kind of density gradient centrifugation and isolation protocols. Then there is isolation of PM using a discontinuous gradient. Then you can use it for isolation of PM by sedimentation through a preformed gradient, that is for 1 to 3 hour spin. Then detergent free purification of caviole, and then purification of detergent resistant lipid wraps. So within your plasma membrane also, like you can isolate your complete plasma membrane, and then again within your plasma membrane you can separate different components like your cavioli or your lipid wraps. So your higher level of separation is also possible using the same OptiPrep. Then purification of lipid wraps in the absence of detergent. So this was detergent resistant and this was absence of detergent. So there will be certain lipid wraps. If you want to see the function of those lipid wraps, how they help in maintenance of your plasma membrane, or if you want to see its interaction with any other external molecules, so you want to isolate that. So even in that you should know whether your particular type of lipid raft is resistant to detergent or not. So if you put a detergent sensitive lipid raft into a detergent isolation method, then your lipid raft will get degraded. So in those cases you have to go with purification in the absence of detergent. So these kind of small details in the protocols should also be checked very efficiently. So then this was first to be developed and involves an isolation of caveoli from a sonicated total plasma membrane fraction. This strategy has also been used for lipid rafts, and lipid raft rich in microdomains renders relative insensitivity to detergent solubilization. So that is what. So separation in detergent containing iodosanol gradients has also been done. So for this kind of different detergent free, detergent plus, lipid raft, colloidal, silica, there will be again small differences in the product. The name will be same, but the catalog number will be differing. So you have to go through, read the entire documentation, and then check which one is suitable for your isolation, your type of isolation. The next question is density gradient centrifugation is based on what? A. Only buoyant density. B. Only sedimentation rate. C. Both A and B. And D. None of the above. That is not no, neither A nor B. What is the answer for this?
I think both A and B, ma'am.
Yes, very good. So your density gradient centrifugation principle is based on both your buoyant density and your sedimentation rate. So it is both A and B. So what is your sedimentation rate? How it affects us? It separates the particles based on the sedimentation coefficient. And then in buoyancy, your density gradient centrifugation, it separates the particle based on their buoyant density. That is, the particles sediment until they reach a layer with the same density as the particles, at which point they stop moving and they can be separated. So like we saw in the beginning, the rate zonal and the isopycnic, in one it was time dependent and the other it was time independent. So there, why it was time independent is because it was getting separated because of the density. So this density is your buoyant density, and it, why it is time independent is the particles, when they reach the same density, then they stop moving. Even after that, you keep centrifuging it at a particular RPM or something. Unless the RPM is changed, the sedimentation, the particle will stay in the same zone. So then you can easily collect that zone and then again purify it from that zone. Then DGC uses the density gradient, which is liquid medium that increases the density from the center of rotation. So this is a centrifugal rotation. So from the center, your density will start changing to the outside. Then the density gradient is created by layering solutions of decreasing concentration in a centrifuge tube from the bottom. So from the bottom, when you start, you will decrease the concentration. You will put the highest layer at the bottom, then you will decrease. Like for suppose if there is 50% density in the bottommost layer, then above that you will put some 40, then 30, then 20. So you will decrease it from bottommost. So when you layer, you will, the first one which you put will be your highest layer in terms of your uh, density. So in previously they asked the question in reverse way. They asked from top to bottom. So that time it will increase from top to bottom. But when you are layering, you will always start with your highest layer of your density. And then your sedimentation rate, as they said here, it is based on the sedimentation coefficient of the particles. So what is the sedimentation coefficient? It is a measure of movement of the particle, that how fast the particles move within the that centrifugal force or that centrifugal speed. And it is also the unit for that is S. Like you can see that the usually the ribosomal RNAs will be denoted as either 16S or 18S or 25S. So that S in that is your sedimentation coefficient. So that S is a Swedberg unit. So that is your sedimentation coefficient, and that is how, based on that coefficient, your that 16S, 18S, 25S were also separated, and based on that only they got to know that these are the different types of RNA in that. So this will also uh, help you know, like like you can relate it with your mass or your shape of the component in the medium. Then yeah, that's it for this. Then the next question is, dash motor neurons regulates size of your muscle spindle or
Ma'am,
intrafusal fiber. Yeah. Tell me.
what is buoyancy, ma'am? What is the difference between normal density and buoyant density?
So buoyant density is based on the, like, in a medium. Like, what is your buoyancy actually? Your buoyancy is nothing but the pressure that your liquid, like when you put something in a medium, liquid medium, the force that your medium puts on that particle, or that solid material, or that molecule that is there in that medium, the liquid pressure on it is your buoyant force. So it will usually be pushing it up, right? Like the you when there is more buoyant force on a particular molecule, it will push the object or that molecule to the top, to the surface of the liquid. Why does it come? Like it floats, right? Certain objects, when you put it inside, it floats. It's not only depending on the weight. Even very heavy objects can also float on the water. Why is that? That is because of the buoyant force that is affected on that object by the liquid or by the water. So it is based on your density of that liquid, that buoyant force will change. So that is your, if when you take your density with that buoyancy property of that particular liquid of on that object, then it will become your buoyant density.
Okay.
Am I clear?
Okay. Okay.
Or do you still have doubt?
Okay, man. I'm okay with this.
Clear, right?
Yeah, I see.
Okay. So basically, you should remember that how it floats, like how different objects can float even if they are heavier. So it is both a parameter of your weight of the body, and then your force that is exerted from the liquid. So let's move on. Next question is, what is the motor neuron that regulates the size of muscle spindle or intrafusal fiber? A alpha, B beta, C gamma, D delta. What is the answer for this?
These questions I'm not sure about this one. I don't know.
Okay, no problem. So the answer for this is gamma. So there are different motor neurons, all alpha, beta, gamma, delta. So each one location is slightly different. Few will be in your intrafusal fiber, few will be in your extrafusal fiber, and then few will help in maintaining the shape, few will help in maintaining the length of it, like that. So here what regulates the size of the muscle spindle intrafusal is your gamma. So your gamma motor neurons are also known as gamma amines. They regulate the length of the intrafusal muscle fibers in the muscle spindles. So what is the main function of your gamma motor neurons is to regulate the sensitivity of your muscle fibers to stretch. So you know, right, when your muscle fibers, when you move your muscles or even your normal body, the muscles start moving and they will contract and then they will relax. So that time they stretch and then they go back. So this gamma motor neurons help in its ability to stretch and relax properly. So it is sensitive to that too, in particularly to the intrafusal muscle fibers only. And how they do this is they adjust the tension in the fibers, like like if you take a rubber band and you pull it, you can feel that tension in the rubber band, right? There will be a pressure there, that is called as tension. So your muscle is also similar to that when it's contracting and relaxing. So this gamma motor neurons will adjust or regulate the tension in the intrafusal muscle fibers, which will help to regulate the length and tone of the muscle. Okay, so because once they contract and relax, they have to again go back to their proper length. So it cannot stop in its contracted place, or it cannot stop once it is completely stretched. It has to maintain its length. And it has, how will that length be maintained? When there is enough tension that is regulated to maintain that particular length in that fiber. So this will be done by your gamma motor neurons, and these are located in your in these are located in your intrafusal muscle fibers in the muscle spindle. And then they are important in your stretch reflex mechanism. Like I said, once they stretch, they have to go back. So that is nothing but your stretch reflex. Reflexes are something that you get involuntarily. So this has to be maintained dynamically all the time. So that is what your gamma motor neurons helps, because it gives that sensitivity in signals to the muscle fibers. Then they work with alpha motor neurons or your alpha MNs to maintain the muscle length and its velocity. Then the proportion, that is the amount or the number, it percentage it contributes, is it makes up almost one third of all the motor neurons in the body. So one third of all the motor neurons is your gamma motor neurons. So here you can see this is your muscle fiber, and this is your intrafusal muscle fiber, and these are your extrafusal muscle fiber. So here you can see you have your alpha amines, and then in that again you have 1, 2A and 2B. So each one has been for a different purpose. So your alpha 1 is your slow twitch fatigue resistance, and then your 2A is for your fast twitch fatigue resistance, and 2B is for the fast twitch fatigable. So your 2A 2B are more similar. It is for your fast twitch response, and then your 1 is for your slow twitch fatigue resistance. This is present in your extrafusal fiber, whereas your gamma is present in your intrafusal fiber. So you can see this is the muscle spindle, this is your gamma. Then this will be again connecting to a network over here, which will regulate and send signals when to relax, when to contract, and how much tension has to be given there to hold it in the particular length. Okay. Okay, that is it for gamma. So next is, what motor neuron regulates the size of extrafusal? Till now we were talking about intrafusal, now we are talking about extrafusal. So what is the answer for this?
It is alpha.
Yeah, very good. So in the previous picture I showed you, in your extrafusal you have your alpha motor neurons. So your alpha motor neurons are large motor neurons, and they innervate in the extrafusal muscle fibers, which are responsible for generating muscle tension and maintaining postures. So basically the function that they do is the same, or it is similar, but it is the location. Your extrafusal requires a different set of neurons, and then your intrafusal will require a different set of neurons. That is your alpha and gamma. So these are located in your spinal cord's anterior horn region, uh, anterior horn gray matter, and your ventral horn. So you know your uh, brain, when you spinal cord, when you give the transfer section of it, this is how it will look. So this is your uh, ventral horn, and this is your anterior horn. So this is present in the anterior horn gray matter. So it will be present in the gray matter, and this is your ventral horn. So you can see here, here they have specified the region where gamma is, because gamma is in one third, it contributes one third, it's in a large number. So you can easily spot that. So here in the same ventral horn, you will have your so in your ventral horn, you will also have your alpha, and also in the anterior horn gray matter. So this is your muscle fiber. So this is like a collateral view they have shown. It will not actually present like this. So this is zoomed in portion of your muscle spindle. So here you can see within your muscle spindle, you have your regular fibers, muscle fibers there. And then this is how they will be connected from your spinal cord to your muscle fibers. Then how do they work? So they receive input from your motor cortex. So this will be in your spine, your motor cortex, and the spinal interneurons. So your neurons within your spine and from the cortex of your spine, they will receive your input or your signal, and then they and from the also from the process synaptic inputs produce an action potential. So you know the action potential of neurons, that is how they transmit the signals. And propagate the action potential down the axon to the muscle cell. So from here, so it's a long way. What trying to say is from your spine to every muscle. It's not only like whatever is close, every muscle in your body will have this, and to each muscle fiber there will be different set of these neurons that are being connected and spread out. So depending on which muscle is being activated at what point, that signal will be sent from the spine and the interneurons in the spine, and those particular set of motor neurons will carry the action potential and it will propagate it to the particular group of muscle fibers. Okay, so it's a very intricate and a very detailed and um, very uh, efficient system. Otherwise, if there is any error in the system, then your motor sensation, your movement, your locomotion, all this will get affected. Then what they release, so this action potential down the line, how this signal is getting transmitted is by the release of acetylcholine. This is your signaling molecule at the neuromuscular junction. So till here it is your action potential, and from your axon to the muscle, the junction where your neuron will now meet your muscle, it will release the acetylcholine. So that is why this concentration is also very important to be regulated, to know how much the contraction and relaxation should occur. And then this will stimulate your muscle contractions. And what do they provide? They provide myotropic factors. So your myotropic means your muscle factors. So these will be sent, or these factors will be attracted by your muscles, which are nutritional substances that keeps the muscle healthy and direct the muscle fiber differentiation. So when your muscle fiber, if it has to, during the developmental stages, when they have to become different types of muscles, then all these myotropic factors will help in its differentiation. And then during the growth, if there has to be generation of more muscle fibers, then also these factors come into play. So this is about your gamma. This was the previous one was your gamma, and this one was your alpha. So your gamma was for your uh, intrafusal, and your alpha was for your extrafusal. In alpha, again there are types, your 1, 2A, 2B. Okay. So now the last question. Dash clustering occurs on myotubules at the neuromuscular junction. A, oh, the answer. A, nuclei, B, acetylcholine receptor, and C, cytoplasm, and D, astroglia. What is the answer for this?
Acetylcholine receptors.
Yeah. So the answer for this is your acetylcholine receptors. So these acetylcholine receptors will cluster at your myotubes at the neuromuscular junction. Like the previous, we spoke that there will be release of your acetylcholine. Here we discussed, right? Your acetylcholine will get released at your neuromuscular junction. So after release, how will the acetylcholine continue sending the signal, or downstream, how will it tell your muscle to contract? So for this reason it needs your acetylcholine receptor. That is why there is your clustering at the neuromuscular junctions of for this acetylcholine receptors. So your acetylcholine receptors cluster on myotubes at the neuromuscular junction as a part of neuromuscular junctions development. So what is the distribution? Like we said, it is in the myotubes. It is initially spread out, but in the adult muscle, they are concentrated in the postsynaptic membrane. So it differs because in your growing stages, you need them at all the places majorly, because your entire body has to grow. But then later it will be, once you have attained your adulthood and the complete capacity of your body, then it will be maintained at particular places where there is more requirement for it, where which muscles you use more. And then if you work out or if you're trying to build muscles, then again the distribution will depend according to your activity. Then your clustering of your acetylcholine receptors is a key event in the development, and it is caused by activation of muscle specific kinase by agrin, a heparin sulfate proteoglycan released from the motor axon. So for this clustering, it also needs the agrin. Agrin is a heparin sulfate proteoglycan. So proteoglycan is your glycated protein that which is heparin sulfate. So this is agrin, and this will activate your muscle specific kinase. So you know all your most of your signaling processes downstream will happen by either phosphorylation or dephosphorylation or ubiquitination in case of degradation and all. So when these kind of signal has to be further downstream processed, you need your kinases. So your kinases are the ones that will phosphorylate your downstream proteins, either your adapter proteins or your signaling proteins or your transcription factors. All your phosphorylation events will be done by your kinases. So the particular kinase here is your MuSK or your muscle specific kinase, which is activated by your agrin. Next, the clustering mechanism. The clustering processes involves the redistribution of acetylcholine receptor proteins and the synthesis of acetylcholine receptors at the synapse. So the synthesis occurs at the synapse, and then the proteins are redistributed at wherever point they are required, wherever the acetylcholine is released. Then the multi-domain scaffold protein RAP-SN is essential for clustering of acetylcholine receptors and forming this. So then uh, everything that happens is not a singular event. It's not dependent or on one protein or one signal or one neuron. It's a collective network of many things that happen. So in the same way, this clustering also requires different things. It requires the heparin, it requires that heparin sulfate agrin, and also it requires a scaffold protein. So the scaffold protein will help in the proper cluster formation of your acetylcholine receptors. It cannot go and just cluster randomly. It has to be in its functional form, it has to be exposed, it has to be available for your acetylcholine to come in, sit on it at its particular complementary region. So that region should be exposed. So for this it needs a particular arrangement. So all this will be done by your scaffolding protein. Then vitamin D signaling. Vitamin D signaling regulates your neuromuscular maintenance and improves locomotion in mice. And it also enhances agrin-induced acetylcholine clustering in myotubules. So many other trace elements and your micro excuse me, micronutrients like vitamin D is also essential for this clustering. Neuromuscular disorders in which this is involved is when there is mutations, or then there are there are auto antibodies that are developed against the components of this signaling complex. Then your neuromuscular disorders will be formed. So when there is mutation in these particular proteins, like this protein, it is important uh, in both the signaling molecule, that is your acetylcholine, and then your other protein is your receptor. So they need to have, they have a function and they have a structure. So when there is mutation, obviously the structure and function, either of it will be lost, because of which the uh, signal will not get further transmitted, and your motor response will not be there, your movement will not happen, your contraction will not happen. And then autoimmune disorders like self generated antibodies. If your uh, body treats your internal proteins itself as a foreign object, it identifies it as a foreign by mistake, due to some other defect, then it will generate auto antibodies against your own body. So that time also these signals will not get transmitted down the stream. So that is how you these clustering is also uh, linked to the neuromuscular disease, because your mutations will also disrupt the structure. So when the structure is disrupted, its maintenance at a particular place or its distribution to the particular place, all this is signal oriented only. When it has to go to a particular place, then if the structure is altered, the clustering also will get altered. And like I said, it might cluster in a way that the necessary region that has to be open for attachment of the acetylcholine might not be exposed. It might be reclustered in somewhere at the unexposed region at the center of the cluster somewhere. Or it might form aggregates instead of a particular arrangement in the cluster where it is actually functional. It might just randomly aggregate in a clump. So that time also it won't be functional. So that is how mutations can affect these receptors very easily, and then it will lead to neuromuscular disorders. So we are done with the questions. Do you have any doubts in all these questions, or if any part you have not understood, I can repeat again also.
Ma'am, is it okay if I ask questions from the previous week assignment?
Previous week assignment, yeah, sure. Which week?
Ma'am, actually, the I think it is week 5. I want to know about the xenophrene media. So where it is
Xenophrene media.
used.
Okay, 1 second. Let me just go to that file. Okay. Yeah, what is your question? I let me find the file. By the time you can ask me what the question is. Hello? Are you there?
Yes, ma'am. Yes, ma'am.
Yeah, yeah,
I
you
just
tell me
want
the
to know.
question.
Yeah, I just want to know about the xenophrene media and the uses where it is used.
Oh, you want to know about it.
Yes,
Okay.
ma'am.
Yeah, yeah.
It is
Okay.
actually week 5 assignment.
Yeah, yeah. I
Yes.
lost file on. Yeah. Yeah, week 5, right? This one. Okay.
Yes, ma'am. This one.
So do you have a question, like a doubt in this, or in this question, or you just want to know what it is, xenophrene media?
I just want to know what it is and where it is application, like where it is used.
Okay, so your xeno-free media is nothing but when you do something with completely using components related to the same species which you're culturing. It's a culture media, right? So usually xeno-free media is used to xeno-free. Xeno means your species specific. So when you're culturing human cells, we use many components like your media. So in your media, if you see, I don't know if it is there here. Maybe it's in your previous assignments. So in your media, we have several components like your sodium pyruvate, your glutamine, your glucose, and one of the major component that is required to grow your cells is your serum. So the serum we usually, even though we are growing human cells generally in normal media like your DMEM media or your RPMI media, these are commonly used media to grow many types of suspension and adherent cells. So here when we add the serum, usually we add fetal bovine serum. So your fetal bovine serum is from your calf, your cow. Okay. So it's a completely different species, right? So when you derive the serum from that species, there might be some components in that species which are not, you know, uh, which are not there in your species, which are not there in humans. So that time those components will become your species strange components. So more, if you're not doing a very specific study related to your highly specific antigen properties or your immunological studies, so these kind of studies, if you're not doing, if you just want to do some basic cell biology work or some other signaling you're studying, so immunology doesn't even play a part, or you're not trying to differentiate your cells into a particular type of stem cells, and you're not, it's not directly translational. It is not that you will make these cells and you will inject these cells into a patient, as a treatment method. When you're not doing such kind of studies, clinical or direct translational treatment studies, then these kind of media is fine. It will not give any major problems or uh, different results in your experiments. But when you're doing such kind of experiments where you have to use these cells to further like explant studies, like you take some part, some tissue from the original plant, you want to do some gene editing in that, and then again you want to put this back into that organism, either it is a plant or a human. Mostly it will be human if you're doing that tissue uh, planting by genetic editing and putting it back into the body. So when you, that time you have to use this xeno-free media, because like I said, when we use this animal components like this bovine serum or anything that is extracted from any other species, you might never know, these might generate some kind of antigenic reaction in the human after the transplant is being done. Okay, then it will cause your tissue rejection and your graft rejections. You might have heard these terms. So these things occur when such kind of growth is done in non-xeno free media. So when you're doing it in xeno free media, you're making sure that it does not have any component that is coming from a different species, so that it will not create any antigenic response or rejection kind of problems in your experiments, in your tissue, organ, etc. Okay, that is the main purpose of xeno free medium. So here you can see in cell culture, human cell lines are cultured using human derived components like human serum. So every time, even for your normal studies in which you don't require such kind of attention, you cannot always, take the human serum. It is very expensive method to extract that, and then the source is also very limited because you can't extract it randomly from a human. Only when it is absolutely necessary you will use this. And then when you make a media which contains everything related to that human species only, then it is called a xeno-free medium. Okay. But at the same time, in this question, what they are saying is you can have some components like some recombinant materials or some DNA sequences which you want to integrate. You can take it from a different source, like from plant or bacteria or yeast, but it should not be animal related, because the DNA sequences will be different for eukaryotes and prokaryotes. So your plants, your bacteria, your yeast will have a particular code of DNA sequence which will affect only in their cells. So, like if you want to overexpress some protein also, generally, what you will do, you will overexpress them in E. coli bacteria. So that time what you will do, you will take the sequence from humans, and then you can't just directly put that sequence into a vector and inject it into your E. coli, clone it into your E. coli. You have to do a step called as codon optimization. So what this codon optimization will do is, the differences in your nucleotides between your eukaryotic and prokaryotic, so that changes will be made in your sequence now. So that is nothing but, to tell it in simple terms, your human sequence will be made compatible for your E. coli to be expressed in that, because your ribosomes, your DNA sequence, your machinery are slightly different in your plant and bacterial system than your animal system. That is why when you take your DNA sequences, you can take it from plant, bacterial, yeast, but you can't take it from animal, because in all animals these systems will be same. So the DNA sequences will affect your human when you take it from animals. But at the same time, because the machinery is different in plant and bacteria, you can take it, purify it from them, and use it in your human sequence. Okay. Unless you do not use any pathogenic kind of bacteria which are safe for humans. So that is what they are saying here in D. And the other things are many things that are processed and unprocessed materials from human sources. Anyways, you need everything from human sources only. And then in B, what they are saying is it does not contain recombinant materials made from non-human animal DNA sequence. That's what. Actually, B and D are the same thing. In the D only, you can say it may contain from plant, bacterial, yeast, but not from non-human animal. So that is similar thing. And then it does not contain ingredients derived from non-human, like this fetal bovine serum directly derived from non-human animals, you should not use in your xeno-free media. And then your animal derived component. But at the same time, what they are saying is you can take some materials from very specific or very highly required things if it is mandatory for your experiment, and it will only be in some non-human animal. You can take it from there, but that has to be processed. That cannot be used as a primary raw material. Okay, it should go manufacturing process of primary, secondary, tertiary level, where they will make it in such a way, or they will modify it in such a way that it will not create any antigenic response in your human cells. So that is what is your xeno-free media. I hope it's clear.
Yes, ma'am. It is pretty clear.
Okay, very good. Even if you have any previous week doubts, you can ask me, no problem. So do you have any
Okay, I
other
have
doubts?
one more doubt, ma'am.
Yeah,
Like,
tell me.
it was not from the assignment question, but my doubt is like, we are dealing about trypsinization, right, ma'am? Trypsinization.
Yes.
In that, we have two different techniques like warm and cold trypsinization.
What techniques? You're not audible.
Trypsinization, ma'am.
How? In trypsinization?
Yeah, we have two methods now, ma'am. Warm and cold.
Warm and cold, yeah, okay.
Yes, ma'am. So, for what purpose it is preferred?
So you're
Like, is there
asking,
any specific condition in which we could prefer one over the other?
okay, your voice is breaking. So basically, I think you want to know the difference between your warm and cold trypsinization. So your warm trypsinization is usually done at
Yes, ma'am.
37 degrees,
Yes, ma'am.
and your cold trypsinization will be done in your 4 degrees, and that will be done in overnight. So basically
Okay.
it is like to,
So
for
is there any
viab
specific condition
yeah,
in
yeah,
which we could
so
prefer to?
it is for like viability of your cells, mostly this cold trypsinization is done. Sometimes what happens is your trypsin is highly active at 37 degrees, okay? So that is why we put it in 37 also to activate it to its maximum capacity. So when you add trypsin, even at room temperature, if you leave it for a long time, it will still function. It's just that it will take some more time to function to detach the cells. But why we keep it in 37 is to activate it to its maximum capacity, so that within 5 minutes or so it will get activated. Okay. So when it is activated in maximum capacity, it will very efficiently start cleaving all the attachment proteins. So like maybe if I don't know if you have attended in the previous leave where there were trypsin related questions, I have told that trypsin is proteolytic. So if you leave it for some more time, it will also damage the outer proteins of your cells, and it will cause cellular damage also. Okay, at 37 degrees. And that is why we leave it to a very small time, and then we take it before it damages the cells. But sometimes maybe there might be certain cells which are very sensitive to trypsin. So those cells might be damaged by trypsin at 37 degrees if it is functioning at its maximum capacity. So that is the reason to reduce that damage level to the cells. Sometimes people do the cold trypsinization, because at 4 degrees it will not function as efficiently as it will be doing in 37 or even at room temperature. So the cold trypsinization will ensure that you have more viable cells, and your cell is with like least amount of damage because of trypsin. So that is the main thing between cold and warm. But it will take much, much longer time in cold trypsinization.
Okay. So because of the time constraint, we are not going opting for that one.
It is not only the time constraint, but sometimes, some cells may not require this. Some cells may have some heat shock stress also if you put it at 4 degrees. So usually that is not preferred normally, unless it is very important for the cells. Like I told you, if there are like really sensitive cells, and you want like zero minimal damage, then only you will prefer your cold trypsinization. Otherwise, most of the times we go with 37 degrees within a short period of time. It will not damage the cells, and it will retain it. Unless your cells are very sensitive, you will mostly do warm trypsinization.
Okay, ma'am. Ma'am, is there any alternative proteolytic enzyme which you suggest other than the trypsin?
Yeah, there is one more. um, I'm unable to recall the name, but there is one more other than trypsin also. It is a little uh, less sensitive than trypsin.
Okay. So until now, there is no such enzyme which is overperforming than trypsin.
It is acutase. Acutase is also another enzyme that is used
Okay.
for in like an alternative to trypsin.
In which cases, ma'am, we would prefer acutase than trypsin?
That is again depending on your cell type, and depending on your your lab. If you do particular types of cells, some people prefer acutase over trypsin. Acutase is little lesser sensitive than trypsin, so it will not damage your cells if they are sensitive. So it's like how why you do cold trypsin, same way at normal temperature, if you want to do, you will use acutase. So it all depends on what type of cells you're using, and then also what experiment you will be doing next. So that also depends. Like if you want to do, like, trypsin detaches your cells outer proteins, right? Maybe you want to check that proteins only, which your trypsin is attacking. Then you can't use trypsin. So in those cases, you will go to acutase, like your surface marker studies or something like that. So based on your cell type and your experiment type, further what you're going to do downstream, you will choose make this decision between acutase or trypsin or warm or cold trypsinization.
Okay, ma'am.
Okay. Any other doubt?
Okay, ma'am. No, ma'am. No, ma'am. Thank you so much.
Okay, okay. Very good. Okay, then I think we can end the session.
Yes, ma'am. Thank you, ma'am. Thank you
Yeah.
for clearing my doubts.
Yeah, yeah, no problem. Anytime.
Sure. Thank you, ma'am. Okay, ma'am. I'm leaving the meeting.
Yeah,
Thank
yeah,
you.
you can exit the meeting.